Welcome to this lecture on synaptic transmission. Have you ever wondered how a thought in your brain travels all the way down to your toes to make them wiggle? Our nervous system is a vast network of billions of cells. But surprisingly, these cells do not actually touch each other. Today, we're going to explore the beautiful and highly regulated process that allows these isolated cells to communicate across microscopic gaps, making every movement, memory, and heartbeat possible. To begin understanding this process, imagine you are standing on one side of a rushing river, and your friend is on the other. You need to send them an urgent message, but you cannot shout over the loud water. Instead, you write your message on a fleet of paper boats and push them across the water for your friend to catch. In your body, your nerve cells, known as neurons, face a very similar problem. They are separated by a tiny microscopic river called the synaptic cleft. Because electrical signals cannot simply jump across this physical gap, the first neuron must use chemical paper boats to pass the vital message along to the next cell. Now that we have our river analogy, let us look at the formal biological process. When an electrical signal, known as an action potential, reaches the very end of the first neuron, it triggers a remarkable transformation. The neuron releases specific chemical messengers, called neurotransmitters, directly into the synaptic cleft. These chemicals rapidly diffuse across the microscopic space and bind to highly specific receptor proteins on the surface of the second, receiving cell. This crucial binding event is what translates the chemical signal back into an electrical response or a physiological action, allowing the message to continue its journey uninterrupted. Understanding this biological process is not just academic. It has profound real-world implications. Without synaptic transmission, your brain would be completely isolated from the rest of your body, rendering you unable to move, feel, or think. Every single memory you form relies entirely on these chemical messages. Because this step is so critical, many medical conditions and pharmaceutical drugs target this exact process to alter human physiology. For example, antidepressants work by keeping happy neurotransmitters in the synaptic gap for a longer time, amplifying their effect. Conversely, painkillers work by blocking the transmission of pain signals at these exact junctions, providing relief. Before we move on to the mathematics, let us visualize this process with a detailed diagram. Here, you are looking at a classic chemical synapse. At the top, we have the presynaptic terminal. Notice the downward arrow representing the incoming action potential. This electrical spike causes calcium ions, shown as blue dots, to enter through specific channels. This influx forces the synaptic vesicles, those circular bubbles filled with red neurotransmitter molecules, to fuse with the membrane. You can clearly see the red molecules spilling into the synaptic cleft, which is the gap in the middle. Finally, look at the postsynaptic membrane at the bottom. The red neurotransmitters bind to the green receptor proteins, opening them up and allowing a new flow of ions to enter the receiving cell. Now that we can visualize the vesicles releasing their contents, we need to understand how scientists measure the strength of this message. They use a concept called quantal release. As we saw in the diagram, neurotransmitters are not just floating freely. They are carefully packaged into tiny biological bubbles called vesicles inside the neuron. The key insight here is that each vesicle contains a standard fixed amount of chemical. We call this fixed amount a quantum. By calculating exactly how many of these standardized packets are released into the gap, we can accurately predict the strength of the resulting signal in the receiving cell. This concept of standardized packets leads us directly to a simple but powerful mathematical relationship. The formula for quantal release is m equals n times p. Conceptually, this just means the average number of released packets depends on how many are ready to go, multiplied by their chance of actually going. Let us break down the symbols. The variable m is the mean or average number of vesicles that actually get released into the synaptic gap. The variable n represents the total number of vesicles that are docked and ready at the edge of the cell. Finally, p is the probability that any single docked vesicle will release its contents when an electrical signal arrives. If the probability p increases, perhaps due to more calcium entering the cell, the total released vesicles m will increase, making the signal much stronger. Conversely, if a disease reduces the available vesicles, n, the overall message becomes dangerously weak. Now that we have our formula, let us see this with a concrete example. Imagine a motor neuron preparing to send a signal to a muscle fiber. We are given that it has 200 vesicles docked and ready for release, so n is 200. When the action potential arrives, the probability of any single vesicle releasing its contents is 25%, so p is 0.25. We are asked to calculate the expected number of vesicles released. We choose the quantal release formula because it directly relates available vesicles and probability to the final output. We substitute 200 for n and 0.25 for p. Multiplying these together mathematically represents the biological reality that not every docked vesicle will successfully fuse. We are finding a specific fraction of the total available pool. The final answer is 50 vesicles released per action potential. Physically, this means we can expect about 50 chemical packets to cross the gap, which provides a reliable safety margin to ensure the muscle actually contracts. So, we have successfully calculated how many chemical packets cross the gap. This brings us to what happens next on the other side. Once the neurotransmitter successfully crosses the gap and binds to the receiving cell, it acts like a key, opening specific ion channels. This opening allows charged particles, like sodium or potassium, to flow across the cell membrane. In physics, any flow of charged particles is an electrical current, and here we call it a synaptic current. This current is the ultimate goal of the entire process, because its exact magnitude determines exactly how strongly the receiving cell is being stimulated to fire its own signal, or inhibited from firing. Before we calculate this current, we must understand the forces driving it. The amount of current depends on two main factors. First, how many doors are open? We call this the synaptic conductance, which increases when more neurotransmitters bind to receptors. Second, how hard are the ions being pushed through those doors? This electrical pressure is determined by two voltages. The membrane voltage is the current electrical state of the receiving cell. The reversal potential is a specific balancing point, the voltage where there is no net flow of that particular ion. The difference between the current membrane voltage and this balancing point is called the driving force. It is the actual pressure pushing ions into or out of the cell. This understanding of conductance and driving force gives us our next mathematical tool. We can calculate the exact magnitude of the electrical current using a modified version of Ohm's law adapted for cell membranes. The formula is I equals G times the quantity Vm minus E rev. Let us define every symbol. The variable I represents the synaptic current measured in standard amperes. The variable G is the synaptic conductance measured in Siemens, representing how many ion channels were successfully opened. The term Vm is the current membrane voltage in volts, and E-Rev is the reversal potential in volts. The term in the parentheses is our driving force. If the conductance G increases because more channels open, the resulting current I will proportionally increase. Conversely, if the membrane voltage Vm gets closer to the reversal potential E-Rev, the driving force shrinks, causing the current I to decrease, even if the channels are wide open. Now that we have used this formula conceptually, let us apply it to a real neuron. We are given that a neurotransmitter opens sodium channels, creating a total conductance G of 5 × 10⁻⁹ Siemens. The resting membrane potential, Vm, is negative 0.070 volts, and the reversal potential for sodium, E-Rev, is positive 0.050 volts. We are asked to calculate the resulting synaptic current in amperes. We choose our modified Ohm's law formula because it links conductance and voltages to current. First, we substitute our known values into the equation. We subtract the positive 0.050 volts from the negative 0.070 volts. This subtraction gives us a driving force of negative 0.120 volts, representing a strong electrical pressure pushing sodium into the cell. We then multiply this driving force by our conductance. The final answer is negative 600 × 10⁻⁻12 amperes. Physically, the negative sign indicates an inward current. Positive sodium ions are rushing into the cell, which will excite the neuron and pass the electrical message along. We have covered a lot of ground today, so let us reinforce the most important ideas. First, remember that synaptic transmission is the vital chemical bridge that connects the isolated electrical circuits of our nervous system. By converting an electrical spike into a chemical release, neurons can precisely control the flow of information. Second, we learned that the strength of this communication depends heavily on quantal release, the exact number of standardized vesicles released into the gap. Finally, we saw how those chemical messengers open ion channels to generate a synaptic current, driven by the cell's voltages. This tiny but crucial electrical current is the fundamental basis of how we learn, move, and interact with the world. You now have a solid mathematical and biological understanding of this incredible process. Great job!